started. Okay, started. Yeah. Good evening, students. Welcome to the foundation class of NEET World. Today we are going to discuss the circulatory system part 2. As already we have discussed about the blood and its compounds, and also we have already discussed in the previous class, that is blood composition, and about plasma also we have discussed. Today we will discuss little brief about the RBC and WBC and platelets. Here right. In the earlier class we have already discussed about some information about the RBC. As already we have discussed, the RBCs are what? They do not have nucleus and also they do not have mitochondria, endoplasmic reticulum. So, is there any use if they do not have nucleus, mitochondria and endoplasmic reticulum? Means yes, of course, obviously. See, the loss of nucleus, as already we have discussed, without a nucleus what it is having? Biconcave shape. Absence of nucleus, it is giving what? More surface area. More surface area means more oxygen absorption, and the same way loss of mitochondria. So, loss of mitochondria also what? If there is no mitochondria, the RBC means the own RBC, it cannot utilize the oxygen for its own, means more oxygen can be transported to the tissues. Is this clear? Yeah, right. And no endoplasmic reticulum. So, the RBCs do not have the endoplasmic reticulum also. Because of this, what is the use means? See, the RBC can be transported in the blood vessel in a flexible manner. So, it can transport easily even in the narrow capillary also. Okay. Some more fact about the RBC number. What is that? See, in the newborn infants, number of RBCs are what? Larger or more in number, that is 6 to 7 million per cubic millimeter. So, generally in adult that is how much? 4.5 to 5 million per cubic millimeter RBCs will be present in the blood, and RBC count is lower by 5% during the sleep. Of course, it is not exactly correct one, but the functioning of the RBCs may be little reduced, and the RBC count will be higher in number when we do vigorous exercise, okay, and even when we are in the stressing condition. So, at that time actually we required more amount of the oxygen. For that purpose RBC number instantly increase, okay. And other fact, that is what? People who are living in a higher altitude, even they also have to increase what? Their RBC number. Why? What is the reason means? In the higher altitude oxygen levels will be less. So, because of that reason, to breathe themselves in a proper way, they need more number of RBCs. So, at height what? 4,200 meters height. So, in that time in the humans the RBC number will be increased nearly how much percentage? That is 30%. Okay. And see, the production of RBC we called it is as what? Production of RBC called as erythropoiesis. What we called it is erythropoiesis. Okay. If the RBC number increased abnormally, if the RBC number increased abnormally, that condition we called it is as what? Polycythemia. What we can call it is polycythemia. If the number to the normal, if the number is to the normal, if it is decreased, we call such condition as erythrocytopenia. What we can call it is erythrocytopenia. These terminologies very important. The production of the RBC we called it is erythropoiesis. If the number of RBC abnormally increased, we called it as polycythemia. If the number of RBC is decreased, we call that process is erythrocytopenia. Do you understand, students? Right. Next, the next one is WBC. This WBC only having other name, it can be called as leukocyte. What we can call it is leukocytes, or else white blood cells, white corpuscles, blood corpuscles also we can call. Clear? So, what is the difference between RBC and WBC? So, here as we have discussed, this RBC do not have what? Nucleus. Okay, it do not have nucleus. Coming to the WBC, these are with the nucleus. These are with the nucleus, and RBC is having what? Pigment, that is hemoglobin, and these are WBC, no hemoglobin. So, this is the two variations we can identify. Okay. So, they do not have nucleus, and at the same time they do not contain what, sorry, they do have nucleus and they do not have what? Hemoglobin. So, that is why lacking of the hemoglobin, these leukocytes are colorless or white in color. Yeah, right. And what is the number of WBC? Means comparatively RBC, WBC number is very less, that is 4000 to 8000 per millimeter cube only. Clear? Very less number we can identify, and the WBC is mostly they will have what kind of movement? Means amoeboid movement. So, what is amoeboid movement? Here you can see they can, while they are transporting in the blood capillary along with the RBC. So in this blood vessel, this all the blood is flowing with some kind of pressure. So, because of that pressure, some of the blood cells, means particularly which kind of cells, that is WBC, that can oozes out. So, with the help of what? With forming what? With a false food, forming the false food. This false food we can call it is as what? Pseudopodia. What we can call it is pseudopodia. So, what is happening? Because of the pseudopodia, this some of the cells, blood cells are comes out of the capillary. So, this coming out process we can call it is as what? Diapedesis. What we are calling? Diapedesis. See here, dia means across and pedesis means oozes out. So, based on the shape and other characters, these WBCs are divided into two types. How many types? Two types. One is granulocytes, and other one is agranulocytes. Granulocytes again divided into three types. One is neutrophil, and the second one is eosinophil, and the third one is basophil. These agranulocytes also again divided into two types. One is lymphocyte and the other one is what? Monocyte. What we can call it is monocyte. So, these WBCs are divided based on the shape and other characters, maybe based on the other characters, right. So, why we are calling instead of these WBCs, can we call as white cells or we should call as white blood cells only? So, the accurate word is we have to call them as white cells. Why? What is the reason? Means as we all discussed, because of the pressure from the blood capillary, some of the blood cells are coming out of the blood capillary. So, means what? They are not in the blood, right. Most of the time they are coming, means 90% of the blood cells are, means most of the time they will be in the tissue fluid. So, around the blood cells, what will be there? Around the blood cells, around the blood capillary, what we can identify? tissues we can identify, as we all discussed, because of the diapedesis process, these WBCs are coming out and they are accumulating between the tissue. That means what? Most of the time they are not being with the blood. So, that is why the accurate word to call the WBC as white cells only. Of course, we can call it as white blood cells also, because these WBCs also belongs to the blood component only. Okay. As we have discussed, these WBCs are divided into granulocytes and granulocytes, or we can call it as non-granulars also, or else agranular. Agranular, means what? Absent. Here granular means what? Inside the cells we can identify granule-like structures. Okay. And in the granulocytes, these are having what? These WBCs are having what? Nucleus, and in these granulocytes the nucleus is lobed in structure. What in structure? They are in the lobed form. The nucleus is in the lobed form. Right. First one is neutrophils. How much percentage of the neutrophils are there? Means these number is the highest percentage, that is 62% is there, and the nucleus is, as we all discussed, it is having what? 3 to 4 lobes it is having. Okay. And in the cytoplasm we can identify small granule-like structure. Clear? And it is stained with what? Neutral dye. So, because of that reason these cells are called as what? Neutrophils. And what is the function of these neutrophils? Means they do what? Engulf the bacteria, or else phagocytosis. Means whenever any foreign material or bacteria, virus, if it entered into our body, so the first one, this neutrophils come and immediately eat it. Okay. And that will be destroyed. Okay. And coming to the eosinophils, and what is the percentage of the eosinophils? Means that will be 2 to 3% only. Clear? And whenever in our body suddenly we might feel itching sensation or some allergic condition, at that time whose number increases vigorously, means that is nothing but eosinophil number will be increased. So, these also inside the cytoplasm we can see the granules, and the nucleus is only bilobed. How many lobes are there? Bilobed. 2 lobes are there. So, we called it as bilobed. And see, it undergo with which dye? Means that is eosin dye, or else acidic dye. Okay. Because of this staining we can call it is as what? Eosinophils. And what they do? Even they are also engulfing the bacteria, means phagocytosis they do, and they are secreting antitoxins. Antitoxins means against to the toxic substance. These eosinophils are secreting what? Antitoxic. And mainly when allergic conditions occurred in the body, means the first cell who comes and fight against to that pathogen, means that is nothing but the eosinophil. Coming to the basophil, this number is very less in manner, that is 0 to 4% only. Okay. And here the nucleus is having what? Large, means many number of lobes it is having with the larger in size, and granules are stained with what? Basic dye. It undergo which staining process? Basic dye. Example for the staining, that is methylene blue. Which color it can take? That is methylene blue. So, because of this basic dye only it can be called as basophils. And these basophils are secreting what? Histamine. They are secreting what it is? Histamine. Along with the histamine it can also secrete heparin also. What it is secreting? Heparin also it is secreting. So, histamine it's mainly helps for the inflammation. What kind of function it do? Inflammation. What is inflammation? Means whenever we will get hurt. So, sometimes you might have seen immediately whenever you will get hurt. So, the skin will get swollen. So, that we called it is inflammation. What is inflammation? In the next slide we will discuss very briefly. Okay. So, these histamines are what they are doing? They are dilating the blood vessel. Means the blood vessel will be increased its diameter. So, what will happen because of that reason? So, when it increases its diameter, so more number of WBCs can come, means which this basophils can come, and they can reduce that injured area. Okay. Then coming to the non-granular or else granulocytes, they are divided into two types. So, here in this granulocytes, we have seen the nucleus is in many lobes, but here in the granulocytes, the nucleus is only single, there is no lobes are present. So, it is divided into lymphocytes and monocytes. So, lymphocytes, how much percentage they are available, means that is 30%. Mainly lymphocytes are lymphocytes helps in the defense mechanism. They produce what? Antibodies. Lymphocytes are producing antibodies. Antibodies fight against to the diseases or any foreign particle. So, as we all discussed, so the lymphocytes are very smaller in size, okay, and the nucleus is what? Single nucleus we can identify. They produce what? Antibodies. And coming to the monocytes, the percentage of the monocytes is, that is 5 to 3% we can identify, and the nucleus here we can see that is in the bean shaped or kidney shaped nucleus we can identify. So, these monocytes at the site of infection transform into what? Macrophages, means whenever infection occurred into our body, so they are transforming into as a macrophage. Even they do kill the germs, ingest the germs, means they engulf it, and inside the vacuole the germ will be get killed. Here you can observe, students, that is all these WBCs also produced in the bone marrow itself only. Where they are producing? Bone marrow itself only. So, maybe in examination, if they will ask if they will ask the descending order of the WBC, so it is little difficulty to remember in order wise from the higher order to the lower. Okay. So, for that purpose I am giving a mnemonic, that is never let monkey eat bananas. Here N stands for neutrophils. Okay, N stands for the neutrophils, L stands for the lymphocytes, M stands for the monocyte, and E stands for the eosinophil, and B stands for basophil. What is that? Basophil. Now, here according to the percentage you can see this is 62%. Here, right, leukocytes are 30% and monocytes are how much? That is 5 to 3%, and eosinophils are 2 to 3% and basophils are 0 to 4%. So, in this way easily you can remember the descending order of the WBCs. So, what is that? Never let monkey eat bananas. Okay. Yeah, students, as we all discussed, all kind of WBCs, what they do? What they do, ma? Phagocytosis, and most of the WBCs do the body defense. Defense means to fight against. With whom we are fighting every day? Every day we are fighting millions of the pathogens. We can't, these bacteria are invisible to our eyes, they are very minute in size, we cannot able to identify. Every day we are surviving with the microorganisms, but are we falling ill every day? No. Why? Because in our body the defense mechanism is there. The defense mechanism can be done by this WBCs. Yeah, right. Now we will see what is phagocytosis. as already we have discussed. Phagocytosis means engulfing the microorganisms and destroying it. That process we called it is phagocytosis. Mostly it can be done by the neutrophils. Here you can see this is neutrophil and here bacteria is there. It is taking inside its body. This bacteria is entered into where? Vacuole. This vacuole is secreting what? Some of the enzymes. These enzymes are digesting that bacteria, or else it can be destroyed into pieces. In this way phagocytosis can be done by the WBCs. Yes, as we all discussed, what is inflammation? So whenever you got hurt, so or else you might have fallen down. So after few seconds you feel that that area will be swollen, right? And also particular area wherever you got hurt, that will it becomes little heat and redness also you can identify. The area can be bulged, that can be called as what? Swelling. And also we can identify what? Pain we can identify. So all these actions are occurred means actually there what is happening? What is happening, ma? The action, defense mechanism is taking place, means inflammation is occurring. So what is the use of this inflammation? Means, for example, here bacteria has come, or else you got injured. So because of that some of the bacteria entered, right? So this bacteria, because of this inflammation process, this bacteria cannot be transported to the other healthier cells. Clear? So this is the use of the inflammation. What characters we can identify? That is increased local heat and redness, swelling, pain. Okay. So mainly what kind of cells comes over there means, that is monocytes and the neutrophils. They come to that area immediately when you get hurt, or else pathogens entered, and what happened? The infected area help in destroying the germs and healing the tissue. This process we are calling as inflammation. Okay, formation of antibodies. Just now we have discussed under the agranulocytes. Antibodies are formed by the lymphocytes who are producing, that is nothing but lymphocytes are producing these antibodies. When these antibodies produced means when the toxic substance entered into our body. Against to that toxic substance, these antibodies will be formed, especially by the lymphocytes. Okay, here you can see this is lymphocyte. So this is producing what? Antibody. Antibody is always represented in the Y shape. See, these antibodies are surrounded by this toxic substance and it can be neutralized or maybe the destroyed. So this process is called as what? Formation of the antibodies. So I think you might have experienced about these vaccines, right? When in 2020 in the corona time also everybody has taken this vaccine. What this vaccine is have, we will see in the next slide. See, this vaccine, in the vaccine what will be there? Paralyzed condition or else a dead microorganisms, or else virus can be present in this vaccine. Through the injection, those paralyzed condition germs or else virus will be injected to the a healthy human being. So when they entered, immediately in our body lymphocytes are present, they will get a response, and against to that microorganism or virus they start producing the antibody. So in this way what happened? Antibodies will be produced, and whatever the antibodies produced, they can be stay for the longer time and the disease can be reduced and the human become as immune to that against the disease. So this is the main principle in the vaccination. Even if the live or alive in condition pathogen entered also, at that time also these lymphocytes get react and they start producing the antibodies. So, this is the mechanism. Okay. So, where these WBCs produced, as already we have discussed, RBCs are produced in the bone marrow, right, long bone marrow, what we have discussed in the earlier class, long bone marrow. The same way even bone marrow, the same way even this WBCs also produced in the liver, spleen and the lymph bones along with that the red bone marrow. Okay. What is the lifespan of this WBCs means? That is only 2 weeks. How many days they can be able to survive? That is 2 weeks. What is the RBCs lifespan? That is 120 days. Okay. Very few days, means 14 days only it can be able to survive. After that again this WBC can be produced. So, coming to the neutrophils, these are considered as what? Granulocytes, right? They can live only few hours, right? How many hours? Only few hours they can be live, but their number is what? It is 125 billion neutrophils can be produced per each day. Okay. And as we all discussed, the worn out WBCs and RBCs both are same. Where they are destroying? They are destroying in the spleen itself. Where they are destroying? They are destroying the spleen. So, that is why the spleen is called as graveyard. What we are calling? Graveyard of RBCs and at the same time WBCs also. So, as we have discussed, the production of RBC we called it as erythropoiesis, and here the production of WBC we called it is leukopoiesis. What we are calling? Leukopoiesis. So, if the number of WBC increased abnormally, it causes what? Leukemia. What is that? Leukemia. Okay. If the number of WBCs decreased, that condition we called it is leukopenia. What we are calling? Leukopenia. You may can ask one doubt, as we have discussed, that is what? Whenever infections or allergies occurred in our body, immediately what? Neutrophils number or eosinophils number will be increased. But that is not the situation abnormally, but that number will be get reduced when the allergic condition reduced. Immediately the number count of the WBC also can be reduced. But here, as I have mentioned, abnormal growth. What is that? Abnormally, if the WBC number is increased, it can be considered as the leukemia or blood cancer. We can call it as blood cancer. Then what is the treatment for the blood cancer? Only blood transfusion is the only way to reduce the severity. Alright. Coming to the third formed element, that is blood platelets, and also it is called as what? Thrombocyte. What we can call it is thrombocytes. Mainly the thrombocytes or blood plate cells are helping in the blood clotting. They are help in blood clotting process. Yeah, right. So, what is the shape of the blood platelets means? That is maybe some may be in the oval shape, some may be in the round structure, and these are also non-nucleated like the RBC. And what is their number means? 2 lakhs to 4 lakhs. The number will be 2 lakhs to the 4 lakhs in a healthy adult. And from where they are producing means the same way how the RBC and WBCs are produced from the bone marrow, the same way these platelets also originated from the bone marrow itself only, but from the megakaryocyte. Megakaryocyte means the shape of the cell will be very larger. So, that cell we are calling it is as megakaryocyte. This megakaryocyte undergo the fragmentation process or maybe by the budding process. Okay. So, this will be split into small pieces and each piece can be accumulated or rounded by the cytoplasm and it become as a platelet or thrombocyte. Okay. And what is the lifespan of this blood platelet means that is only 3 to 5 days only. How many days? 3 to 5 days, and after the 3 to 5 days they are also will get destroyed and where they will go and buried means that is also mainly in the spleen itself only. Okay. Where this spleen is located? It is a lymphatic organ, it is located in the abdomen region. Okay. And as we all discussed, it is very important for the blood clotting process. So, whenever injury occur and whenever cut is happened, so immediately there we can see these platelets and from the damaged tissue thrombokinase will get start release. Okay, see here what happened, injury cut occurred over here. So, from this injured tissues and the platelets will come immediately. So, these platelets release what? Thrombokinase or thromboplastin. So, they start releasing the at the site of the wound. What is that? Thrombokinase or thromboplastin. So, this also recently we are calling this is as factor 10, that is start factor. What we are calling it is start factor. So, this thrombokinase in the presence of calcium ions. These calcium ions are present in where? In the plasma itself only. Okay. So, in the presence of calcium ions, this thromboplastin is converting into prothrombin. Okay. This prothrombin also present in where? In the plasma itself only, but it is in inactive form. When injury occurred, when something accident occurred, at that time only when it is initiated, when it get activate, then prothrombin get activate and this prothrombin is converting into what? Activate, from inactive form to the activate form of thrombin it can be converted. Now, this thrombin what it is doing? This thrombin is become active, it act as an enzyme, it is converting what? In soluble form of fibrinogen. This fibrinogen also present in the plasma itself only, but it is in the soluble form. Okay. So, in the presence of calcium, now it is converted into insoluble. How it is converting? Insoluble form of fibrin threads. How? As fibrin threads. In this way, whenever a cut happen like this, fibrin threads can be come and accumulate and finally a clot formed. Now, this clot what it do? It stops the bleeding. What it do? It stops the bleeding. Otherwise, if it is not happen, if this clot is not happen, continuation blood supply can be taken place and it leads to the death. One more time, see the process ma. Whenever injury occur, what happen? See, injured tissues and platelets starts releasing what? Thromboplastin or thrombokinase. It is called as what? Factor 10 or start factor, and this thromboplastin is converting prothrombin into how? Thrombin with the in the presence of what? Calcium ions. Okay. And this thromboplastin converts a prothrombin, inactive prothrombin into active thrombin, and this thrombin converting the soluble fibrinogen into insoluble fibrin threads. So, this is the mechanism of blood clotting process. Okay. So, if the same process if we want to see in the test tube. So, here blood is collected, fresh blood is collected in the test tube. In the blood what will be there? Serum will be there, fibrin threads also present, and the corpuscles means WBCs, RBCs, platelets, all are present in the fresh blood. So, if we will keep the test tube for a while aside, so we can identify a yellow colored liquid like substance upper side, and in the bottom region a clot with the cells we can identify. In this clot what will be there? WBC, RBC, platelets and the clotting factors will be present. Okay. So, what is the difference between serum and plasma? So, the serum do not have any clotting factors, it do not have clotting factor, and plasma is with the clotting factor. So, this is the difference between the, this is also liquid only, this is also liquid only. The major difference is the clotting factor, that is fibrinogen. Clot is a solid part. Here right, see, low platelets, if platelet number decreases, is there any problem to the human being? Yes, of course it is a problem. Why? Because the platelets are mainly helpful for what purpose? For the clotting process. Here right, see, when this platelet count will be reduced, you might have heard that a person is suffering with a dengue fever. Here right, dengue is caused by the mosquito bite, right? It is a, by which mosquito? That is by the female Aedes mosquito. Dengue is a virus, it can be transmitted through this female Aedes mosquito. So, at that condition what's happened? This platelets, means in the condition of dengue, the platelets number will be reduced. So, it causes what? Coagulation may be reduced. It leads to the hemorrhage, means internal bleeding may be occur. Clear? So, that is the dangerous thing which can happen in the dengue patients. Clear, students? And there is one wrong notion, means a wrong misconception is there. That is what? This blood clot may be formed when our wounds are exposed to the air. No, it is not at all correct. So, this is because of the clot is formed because of the fibrin. Okay. And at the same time, sometimes inside the blood vessels also clot forms. See here, this is normal blood vessel. Here the yellow color substance is the fat deposits or maybe calcium deposits. So, it narrowing this blood vessel diameter. So, now these blood cells cannot be passed freely. So, sometimes all these can be accumulated because of this narrow passage, okay, and it leads to the blood clotting. So, cholesterol deposits also causes the blood clots. Yeah, blood transfusion and blood groups. How many blood groups we are having? Already we know that we are having A blood group, B blood group, AB blood group and O blood group. Total 4 blood groups we are having. And when the blood transfusion can be occurred, means if any accident occurs, severe blood loss happen at that time, this blood transfusion we can see mostly the blood transfusion will be with the same kind of blood, means A blood group can be give to the A, and from B only we will take the B blood group, and the AB persons can give to the AB, and O blood group can be give to the O blood group. And you know, the one who is giving blood we call them as donor, the one who is taking we call them as the recipient. And while the blood transfusion is occurring, blood should be matched. What is matching? Same kind of blood group should be present. Yeah, right. So, what are the blood types and what is RH system, ABO blood system? In next class we will be seeing.